Good morning, very good morning, and warm welcome all of you to this distinguished lecture by Professor Sue Lin-Chao. Now I request our Vice Chancellor, Professor Appara Purle, to accompany today's guest speaker, Professor Sue Lin-Chao, onto the dais. Now I request Professor Appara Purle to proceed over the function.
Good morning, everybody. It's a great honor to invite Professor Sue Lin-Chao to University of Hyderabad. Professor Sue Lin-Chao is a highly distinguished scientist working with E. coli, molecular biology of E. coli, if I put it in one line. But she has made several outstanding contributions in the past 2 decades. I've known her very closely. Maybe, it's now 2 decades I have known her. Since 1998, I have known Professor Sue Lin-Chao. She works at Institute of Molecular Biology as a senior professor, and more importantly for University of Hyderabad, she has considered this university as her second place next to Academia Sinica, Taiwan, where she is working. She has visited several times. Not every time she had an opportunity to give a talk, but a couple of times we made her to give a talk, and this is one such occasion that we requested her to give a talk, which is put under the Distinguished Lecture Series of the University. I'm going to request anyway Professor Danan to give more a formal introduction about the speaker, because she has several accomplishments which he is going to mention to all of you. I would like to mention a few things, like as a person responsible at Academia Sinica for international collaborations, she has always put her foot forward to encourage collaborations between University of Hyderabad and Academia Sinica, Taiwan. In these 20 years, I know a good number of our students have gone for different types of different projects at Academia Sinica, Taiwan, including some postdoctoral fellowships. And there are also visits by our faculty from our School of Life Sciences and also from School of Chemistry on different occasions. And we have a formal MOU with Academia Sinica. And School of Life Sciences, to start with, has developed a second international graduate program. First of it was started with Germany, that was Professor Shivkumar was coordinating. And then we have now another graduate program called jointly with Academia Sinica and University of Hyderabad. And she has been instrumental in planning, designing, planning, and then see to it, and finally she saw to it that the program is in place. So we have such collaborations encouraged by Academia Sinica through, most of it has been through Professor Sue Lin-Chao. And therefore, when she considers this as her second home, we also consider her as our own person. But today, this being a distinguished lecture, I am going to request Professor Dayananda to introduce to the audience what Professor Sue Lin-Chao and her academic credentials are to the audience. Professor Dayananda.
Morning to all of you. It is my privilege and honor to introduce the distinguished guest today, Dr. Sue Lin-Chao. In fact, there is no need to have any introduction to her because every one of us is known to her. And when I entered along with her to this auditorium, many people flocked around her and started introducing and exchanging pleasantries. But formally, I wish to put her academic accomplishments before you. Dr. Sue Lin-Chao was born and educated in Taiwan. She received her bachelor's degree in biology from National Changhua University of Education. After receiving bachelor's degree from Taiwan, she moved to do her master's degree and PhD at University of Texas, Dallas. She completed her PhD work on copy number control of coli-1 plasmids and replication and copy number of the plasmids under the tutelage of Professor Hans Bremer. And whosoever teaches copy number control and plasmid incompatibility, it is a textbook matter, and without teaching her findings, it is difficult to cover that particular topic. And after finishing PhD, by doing such a wonderful work, she moved to University of Stanford and worked with in the laboratory of Stanley Cohen. And she is the first to develop mammalian gene trap system using bacterial LAGZ gene, reporter gene. In 1990, she returned to Taiwan and established a laboratory for doing molecular biology work in Academia Sinica. And since then, she has been working on RNA degradation machinery using mammalian as well as E. coli systems, and her keen interest is to know its relationship to the disease. She has won several awards, and to name a few, in 1995, she got selected as one of the top 10 distinguished young scientists of Taiwan. And it is, this award is given by government of Taiwan and through Academia Sinica. In 1997, she won competitive grant and established a very distinguished laboratory at Academia Sinica. And this grant went for 5 years, and during this time, several graduate students and postdocs received training under her tutelage. In 1991, she got elected for Asia Pacific International Molecular Biology Network program, and she held it till recently. And she is also recipient of the Third World Academy Prize given by Third World Academy of Sciences. She is serving currently as a distinguished adjunct professor for a half a dozen Taiwanese universities. And she, of all these things, she is a strong believer of international collaborations, and she was the founder director of international collaborations in Academia Sinica, and she actually moved around the globe and established an international program on molecular biology and genetic engineering. Lin-Chao has authored 73 peer-reviewed publications, and she has 3,500 citations to her credit. And she is also serving as an ad hoc reviewer for Nature, PNAS, RNA, current microbiology, molecular microbiology, genome biology, MGG, JBC, several other top-rate journals. Besides being a top-rate academician, she is a very good friend of University of Hyderabad, and she is a strong advocate to promote the collaboration between this university and Academia Sinica. Several students have gone and graduate to work in her laboratory. Currently, one of my students is working in her laboratory. With these few words, I thank University and our Vice Chancellor for giving me this opportunity to introduce this distinguished guest of today.
Please.
Okay, so let me start at Namaste. Good morning. Dear Vice Chancellor, Prof, and colleagues of UH students, my dear friends, good morning. It's so happy, and it's my honor, great honor, to be invited to give this distinguished lecture. And actually 20 years ago, as Vice Chancellor mentioned, I came to India, and first time I visit University of Hyderabad. So since that time, I cannot remember how many times I have come back to University of Hyderabad. But every time I come, I feel the warm welcome of the university. So gradually, I was turned into one member of University of Hyderabad, working at the academic syndicat, Taipei, Taiwan. Okay, so coming back to give this lecture, I feel very happy and exciting to share the update results with you. So with all this long-lasting friendship, I really would like to take the opportunity to say thank you, namaste, and say shukriya to you all. Okay, so the first, I would like to begin by introduce to you some great scientists whose influences to me significantly on my research interest. So this is the talk I'm going to talk about it. So let's see. I will first, a few minutes, cover the introduction part of my work, and then some of the scientists that I really appreciate the gratitude for their really inspiration, their work. And then I'll touch upon the function, actually it's multiple functions, of the enolase present in the degradosome, then followed by new unpublished result on anaerobic phenotypes of this mutant. And this mutant actually, like that of the E. coli NISO 1917, that is 100 years ago, was discovered from a German soldier, and that use currently is the well-established use for the probiotic treatments. Then some of the direction I'll mention, and that's what the talk I'm going to cover today. So first, I was really taught in high school for 4 years before I moved to the United States to study abroad with my PhD dissertation. So I work on the coli-1 type plasmid, as mentioned by the Vice Chancellor, that at that time I was very interested in how bacteria can control a plasmid copy number, particularly the coli-1 type plasmid. At that time, the real engineering plasmid called PBR322 that was created in 1977, and it's one of the first widely used E. coli for molecular cloning. Okay, and so this plasmid is derived from coli-1 type plasmid, and the replication control was by two non-coding RNA. Primer RNA is called RNA2. The antisense is an inhibitor RNA called RNA1. Okay, and you must know this actually is the first non-coding RNA has a biological function. But however, I want to tell you, if you now look into Google search, you do RNA1, everything comes out is the RNA interference. So you are not longer to find such information. So I would like to mention, and this is the model system that I, at very beginning, to work on my dissertation. And this turnover after RNA1 turns out is very important for maintaining high copy number. So for this, I should mention two great scientists that I really appreciated. So first one is Dr. Junichi Tomizawa. At that time, in my PhD study days, I really follow his papers closely. Every year, he published a paper in Cell, okay, and big paper, and many of the work is really in vitro, elucidate the molecular mechanism, how RNA1, RNA2 interaction determine the RNA2 secondary structure, and that lead to the replication. It's a functional replication primer. So when RNA1 binds to the RNA2, they change the conformation of the primer RNA and then fail to reinitiation of plasmid replication. That is the way he really have great contribution. So I was really not just study his papers. During the journal club, I discussed about his paper with the students and with the faculty. So finally, I brought up this topic to my mentor, Hans Bremer. He is a ribosome person. He's really more GPP stringent control of the gene expression. But I mentioned to him that maybe the plasmid replication control is under stringent control. So he was very pleased to think that I have an idea. Then, okay, go ahead and do it. So I ended up as a dissertation on the physiological control of plasmid copy number. About the same time, that is in 1980, another great scientist is David Perian. And this scientist is in Washington University, WashU, and he is the person very famous for ribosomal RNA biogenesis. And he is the person worked out the 30S ribosomal RNA, this long transcript, actually spliced or processed into 16S, 23S, and 5S, and a few sTRNA, okay? And that's great contribution. And at his late time, he discovered that enzymatic activity, ribonuclease E, is responsible for the precursor of 5S RNA. So they cleave 9S, the pre-5 RNA, to form 5S RNA. But at his time, he was thinking about this ribonuclease E, because discovered is a ribosomal RNA processing enzyme. He anticipated the ribonuclease E role in messenger RNA may be indirected. Okay, so at his time, he was able, I think a year before he passed away, he was able to in vitro synthesize the long RNAseE, but he did not get chance to continue to see the recent progress after he passed away. So at his time, he thought this ribonuclease E maybe indirectly participate in mRNA degradation, but now I will tell you current updated information. Ribonuclease E activity he's discovered, or the gene he's discovered, actually has many important roles in different way. Of course, it is an endoribonucleases, but he can have substrate range from mRNA, transfer RNA, ribosomal RNA to small RNA, and not just work alone. The ribonuclease E work as a complex. Nowadays we call it as an degradosome. Okay, so at that time, as I told you, I was so interested in the RNA1 degradation and the relationship with the degradation and copy number control. So since I have a good experience in studying plasmid copy number and determine the RNA1 half-life, it was so short. So I wrote a letter to Stanley Cohen at my 5th year. At that time, before I graduated, I wrote to him, said I had an idea. I would like to carry out a program that you may be interested in it, because he's a plasmid person. And I want to know how the replication in vivo is controlled or determined by the RNA stability. So that's what I wrote. I sent out the letter. And then one day, there's a story for you. One day, my boss, Hans Bremer, ran out of his office and looked at me. He said, Sue, did you apply your job to people? I said, yes, I sent a few letters, including Arthur Comber and Stanley Cohen at Stanford. And he said, I got the phone call from Stanley Cohen, and both of them was discussed about you, and he told me, he said, Sue, I recommended you go to Stanley Cohen's lab because he's younger. Maybe that's at that time, that's why his comment, because more relevant to plasmid. Then 5 minutes later, I got a phone call from Stan and told me that he is arranged a ticket to send to me for round trip for interview. So I went to Stanford and I gave talk, I discussed, and one day interview, I got a job. So next year, I joined Stan Cohen's lab, and that is the year we joined as a party in his lab. So by being able to join his lab, I first realized that his laboratory has really many projects going on, and I don't know how he could manage it. But anyway, one of the projects at that time, he needed a postdoc to join, is to develop this prototype called gene trapping. And this is now very many people use it for lentiviruses version, but at the very beginning, and we developed, and it's no lack of the promoter. So by transfection into mammalian cells, use like Z as a reporter, you'll be able to identify gene, their expression at a particular stage. And so we isolate many of the genes called GET gene, and currently my lab has a project on GET gene study their function. Okay, so come back to the RNA1. At the same time, I then started my RNA stability program in his laboratory, but I did not finish with the first paper when I returned back home in 1990. And so I continue in the laboratory, the new lab. I finished the paper, and so we was able to publish a paper next year. Since 1990 till now, there are many trainees from my lab. They are working on this project, and some are working on the mammalian GET7. And so a few think Parker, he know him. So he's now in the Himalaya Indian Institute, is a senior scientist. And then Vladimir Kabady, he is now a professor at the Spain Research Institution. And he is now in Singapore, Dimitri, in a biotech company. And the new fellow, and then he's in Hungary, now recently he's got retired. And then there's a new family currently, those are the people are still in my lab, and including Ashang, that he joined me about 6 to 7 months now, by now. And I'm happy with him. He's really a very sincere, hardworking student. Okay, so I'll show you this at the first slide that remind you the old days of this great scientist. He said, once we understand the biology of E. coli, we will understand the biology of the elephant, right, by the French scientist, Giacomo. But of course, now you can replace this E. coli with different organisms of your favor. But I just want to say that I really use this model organism for more than 20 years, almost 30 years, and I'm really very happy with it. This is a portion of the E. coli cell. The yellow is the string of the DNA and covered with this polymerase synthesized. White color is the RNA, and then cover all the ribosome. And this scale, amazingly, is very close to the real cytoplasm in E. coli. So it's very crowded, but it's free, so it's dynamic. And with this, you can imagine this white messenger RNA degradation needs to be really well controlled. Also, the ribosome RNA inside the ribosome automatically they need to get degraded. So how it works? So as I mentioned, all cell, individual, even the mammalian cells, the ribosome RNA, tRNA, messenger RNA undergo multiple cleavages, processing, and eventually will be degraded. Now, these RNAs are nonetheless degraded by many of the ribonucleases. Now, and they are all same, even the same ribonucleases, they have the different substrate. And here I show you about 21 enzymes of E. coli bacteria form. They have a broad importance for the turnover of cytoplasmic messenger RNA. And many of them now actually have a broad role in also stable RNA, small RNA regulation. And so they have endoribonucleases they cleave the RNA from internally, and then exoribonucleases from 3' end to the 5' end. And 5 exoribonucleases, this is not found in E. coli, but actually it's found in the B substances have such 5' end to 3' end exoribonuclease. And interestingly, bacteria has 5' end modified enzyme, such as RPPH, and termed as a capping enzyme. And so they stabilize the messenger RNA for translation. And the 3' end, there are few modification enzymes, such like poly-A polymerase, and they was also jointly discovered by my lab and Stanley Cohen's lab. Bacteria actually has a polyadenylation, and they function as helping RNA turnover instead of stabilization. Actually, it turns out in mammalian cells, the mitochondrial messenger RNA, the turnover is enhanced by polyadenylation at the 3' end. So the mitochondrial version of DNA degradation and E. coli version of DNA degradation actually is very similar. So here I just want to take out of the 21 ribonucleases identified in E. coli, only 3 are essential. Ribonuclease P, that is the enzyme for tRNA maturation. The oligoribonucleases, that is the terminal enzyme to degrade all the nucleotide, RNA nucleotide into nucleotide for recycling. And then ribonuclease E, and that's I'm going to talk about today. Now, bacteria, as I mentioned, messenger RNA, like eukaryotic cells, have poly-A, and that affects mRNA turnover. Also, the PMPS is an exoribonuclease from 3' end to 5' end. It's conserved from bacteria to humans. And you can see the domain structure of this enzyme organization are similar among the kingdoms, okay? And actually the PMPs and dead box helicases, they also form as exosome-like complexes. And those are functions similar to in mitochondria and also in bacteria, right? So I come back to the endoribonuclease E. This is huge, large. Normally the ribonuclease is not as big, but this is 161 amino acid, right? And they have 2 functional domains. The N terminal is a catalytic domain, carry out the enzymatic activities. The 3' end is a platform, is a skillful region that binds to many interacting proteins. And their function, as I said, the catalytic function is the substrate is the mRNA and tRNA for their function. So the first demonstration to show ribonuclease C, apart from cleave the 9S 5S pre-RNA, we demonstrated that this called small tRNA, the ssRNA, actually is cleaved by ribonuclease E at the 3' end, and that leads to the functional as a tRNA. They can rescue the ribosome store or the truncated polypeptide. They can go to the tRNA site and then continue to serve as mRNA. So this RNA is called the tmRNA because function is tRNA and mRNA, and this peptide actually taking and then fusion the protein will then move to the periplasmic for degradation. And it's a quality control. So that is the first evidence to show ribonuclease E also has a function in transfer RNA maturation. Okay, so in addition to process the stable RNA species, here I show you another example is the coding antisense RNA, and that's the joint paper. The first paper now it's become a classic paper when people talking about antisense regulation. The RNA stability has a plasmid control mechanism. So as you can see here, this is a stop transcription. So RNA1 specific probe detected, they got degraded very fast, right? And so if you have ribonucleus E inactivated at the non-permissive temperature, then the stability is got very stabilized. And at this moment, the plasmid replication is inhibited. Okay, so that give the first link of the RNA1 can regulate the replication of CoE1 plasmid in vivo by its stability. Later on, the model RNA has been used by other laboratories to study for substrate specificity. So in the first 5 years, from 1990 to 1995, we continue to use RNA1 and to look for the substrate specificity. And so I'll show you after 1996 how we then discover ribonucleus E actually is not alone. When it's working in the cytoplasm, it works as a complexes. So this is I just mentioned the first 5 years is the target mRNA talking about the substrate specificity. Turns out this ribonucleus E recognizes the substrate with very simple rule. It's single-stranded and AU-rich. So you can imagine single-stranded and AU-rich is everywhere in messenger RNA, in ribosomal RNA. But how can the RNA can maintain its lifespan? Because there are proteins protected from degradation. There are ribosomes protected from degradation, and that's how it works. For small RNA, the protein HQ is the enzyme protected small RNA from degradation, and that now is very clear to the field. Okay, and from here, so we use this ribonucleus E fuse to detect a flag tag, and then we isolate it from the cell lysate, and we discover they are additional to ribonucleus E. They have a PMPS. As I said, this is a 3 to 5 exoribonucleases. Has RHLB is RNA helicases, and also an glycolytic enzyme enolase form as a very stable complexes. And since 1996, you see the binding proteins of the degradosome have been identified. They are including many of the chaperone protein, polyA polymerase, ribosomal, some of the ribosomal proteins, including the one we identified is the L4, and some of the activity regulatory protein they are formed. So it's a very dynamic, but the protein that I mentioned, those 4, are formed very stable complexes. You can purify it all the time. Co-purification is always contain those. And then my PhD students use the EM and immunogo level to look at the subcellular distribution of these complexes. And he discovered that the ribonucleus E is, along with other helicases, actually is really near the inner membrane, right? So those localization is sufficient. Even you use truncate the N-terminal, they have a catalytic domain. So the membrane interaction really determined by the first maybe 500 to 600 amino acid of the N-terminal half of ribonucleus E. So if you now look at the N-truncate one that's here, but if you look at other ribonucleases, or the interacting proteins, if you remove the truncate, only N-terminal, RNAseE is still in the membrane, right? This is when it's only N-terminal. But the other component protein, this is helicases, they now distributed to cytoplasm. But if you ectopically express full length again, right, 161 amino acid, those helicases now can be detected, go to the inner membrane again. So the complexes, including the interacting protein, are localized in the inner membrane. So that is what it started. The field started looking at other ribonucleases in other bacterial system, and the homologue of the enzyme, sure, many of them actually is a compartmentation in the membrane. And that is very interesting. If you're thinking about the slide I show you, there are DNA, many mRNA ribosome, and then these complexes, they are not just ribonuclease complexes, there are many other complexes, and they have their own niche, their own place. So the regulation has become very important, right? And I mentioned also to you, a Prof interact with the ribonuclease E, PMPs and HLB also can form exosome-like as independent complexes. So entire mRNA regulation become very complex and very interesting to us. At the moment, the three-dimensional structure of the RNA-degradosome has not been able to solve, and because the protein is very big and its membrane protein is not easy to be purified. But our lab collaborate with some of the laboratory. We are trying very hard now to see whether we can do it. And this is just to show you the exosome-like complexes. It turns out we demonstrate that at least in vivo, if the mRNA, for example, here is the CISB, is a cysteine regulons, and this is the transcription, the master transcription activator. Their degradation is from 3' N to 5' N. So their secondary structure won't be able to conquer by the PMPs. Therefore, they require RNA helicases to unwind it, become single-stranded, and so PMPs can successfully degrade this CISB protein. If the interaction is interrupted, then the CISB mRNA will be stabilized, and that is the genetic approaches that we use to study the function. Okay, so I'm going to show you, come back to these complexes with everything together. So in 2011, my postdoc was interested in knowing why they had to be in the cytoplasmic membrane. So we wanted to know if we have membrane constitution with this ribonuclease E, what happens. So we did all the biochemistry study, and we demonstrate actually this membrane binding, they can stabilize the protein in terms of their chemical properties. If they're more resistant to thermal, they are higher catalytic activity, they have better affinity to their substrate. And so that tells us how membrane is really good for maintaining the structure of these complexes, and also it's good for serve as a external extrinsic signal comes in, and you can regulate whole subset of classes are for degradation. So that is the one of the continuum of the story after we discover in the membrane. So the challenge is the following. This complex in the membrane, and so do RNA processing and decay take place in the cellular compartment in bacterium? And if it is, then how? And I know some of the scientists in the ribosome biogenesis, they believe the ribosome processing they can detect takes place near the membrane, but they don't know what is the mechanism. And so it's physiological importance of the complex associated with the inner membrane, and also the E. coli naturally is in the adaptation, the growth adaptation to anaerobic niches in our intestinal human and other warm blood animal. So therefore, we would ask a few questions about what is about RNA-degradosome complex in the cytoplasmic membrane, right? And in this, we want to know if this homologous RNAseE, they are all, and the interacting protein are all exist in enterobacteria families, then this family are facultative anaerobic bacterium. And so many members of them are part of the gut flora. And so the host intestine are beneficial, while other exist in animal as a also as a detrimental parasites when you got sick from their food poisoning also. So therefore, we want to know whether this membrane-bound RNA-degradosome play a key role in stabilizing this strain in adaptation to anaerobic niche from anaerobic growth environments. So here is a system, is a laboratory system instead of animal system, just to provide as an anaerobic. And so including the growth chamber is with anaerobic with nitrogen, and you can close it once you get all the oxygen out of the chamber, and so that will maintain the microanaerobic growth condition. And then even the collection and imaging system, everything is under anaerobic condition, and so you can compare in parallel about the culture growth, aerobic and anaerobically, okay? And as I mentioned, this degradosome, in addition to contain P and PS helicases that we know is the exoribonucleases complex, there is a enolase, one of the major components of the degradosome. And this enolase is a key enzyme of glycolysis. They catalyze the reversible conversion of PPG to PEP in the 9th stage of this 10 reaction. So it is a very essential if the bacteria use glucose as a sole carbon source, and under anaerobic condition, all the energy really is only dependent on this. So it's very important. So why this enolase present in the degradosome, and what is the particular function, and what's related to the RNA degradation? So we wanted to address this question, and at that time, it's known that this C-terminal, the member of the interdicting protein had their own individual binding site at the C-terminal. So we were able to design an experiment to remove the interacting specific interaction at 20 amino acid from chromosome. This is not a plasmid. In the plasmid, it's from chromosome. Use a very well-known strategy, recombinase strategy, to take away these 20 amino acids, so block the enolase interaction. So this strand will able to produce this degradosome, but without enolase. So this is called enolase-free degradosome. In compared to the wild type has enolase in the degradosome, and so we will call enolastic degradosome or enolase bound degradosome. All right, that is the idea we would like to do. Okay, and so in order to do another control experiment, this is the area we create another mutant and deleted also 20 amino acid adjacent this site. So this strand, the cleavage site is from 9A4 to 1011. It's also about 20 amino acids. So when you delete it, this strand should have complete degradosome. So it's called enolastic degradosome, as I mentioned. So this strand, wild type, and this strand should contain complete degradosome. But the strand of A23 should contain the enolase-free degradosome. However, you must know, in this, all of those three strands, this is just to mention to you the engineering, a long process to engineer in the chromosome. So it's not a plasmid-driven expression. So those three strands, as you shown here, wild type, control strand, the mutation is at this site, okay? And this is the strand of interest. So first of all, isolate the degradosome, see what happened. So here I show you the wild type has complete 4 components, PMP, RHLP, enolase. And in the oxygen present or without oxygen, they are all very similar. So structure and stoichiometry-wise, in 2 growth conditions, they are very similar, okay? Now, the control strand, 984 strand, you can see that is also complete degradosome. So that's okay, that's good. So those 2 strands has enolastic degradosome. A231 has everything else, but there's no enolase. So this strand is the complex is enolase-free degradosome. However, you must know in all of the 3 strands, enolase is in the chromosome. It's all wild type. It's just the enolase interact or not interact with the RNAseE. All right? So the glycolysis in all the strands, either is a free enolase, bound enolase, they are maintained. They are okay. Okay, so that is the experimental strand that we have. So we want to know what happened. Is anything to do with the ribonucleus E? So this is the data to show you ribonucleus E when you switch from aerobic to anaerobic growth, the level, the amount of ribonucleus E is very little. It decreases. And then you add the chloramphenicol to stop protein synthesis. You see ribonucleus E is stable under aerobic condition. In the anaerobic condition, the turnover rate is very fast. So the conclusion is RNAseE decreases level is due to the degradation of the protein, because northern blotting has shown their messenger RNA are the same. And I don't know at the moment what protease that is responsible for this degradation. Another interesting is remarkable morphology that we observe. You can see here on the aerobic E. coli is a rod shape. When it grow on the anaerobic, it is filamentous, like you see in this picture, okay? And so larger than 70% cells are categorized by a filamentous shape. And so we want to know, can this completely resolve within, if you ship back to, if you supply with the air, would this will be resolved? So that's what we did. And I'll show you. So how do I do? Sorry. So what you see here is a 0 minutes, and it was able to recover. Okay, now you can play. So it takes about 3 hours after shipping back to aerobic condition, and it's completely resolved. So it is dependent on oxygen, right? But I also told you the ribonucleus E level is decreases. So how is the correlation of the oxygen and ribonucleus E? And so we then did experiments. So we want to know if we can acopically express RNAseE under anaerobic condition, whether it would do the same job. So here, just first to establish the ribonucleus E and oxygen relationship. So this experiment, we then do aerobic, anaerobic, aerobic alternative growth. And we look the ribonucleus E protein level. And you can see the protein is correspondence to the oxygen level very well, right? When it's low oxygen, the ribonucleus E is getting less. When it's coming up with the oxygen, and protein gets stabilized and is more protein. And also the filamentous was also completely correlated well with this. So when you have low ribonucleus E level, low oxygen, and they get elongated and come back with rod shape. So if you under this anaerobic condition with no oxygen, but you overexpression RNAseE, and you can resolve the filamentous structure. And this is just to show you. So now you link together. This is a conclusion, can tell you that ribonucleus E regulates the cell filamentous morphology, and it is oxygen level dependent. And that is really a very interesting that we first time observed. But how to go from there? So RNA-degradosome may be involved in this acquiring the filamentous under anaerobic condition. As I mentioned to you, there are many components. So we would then easily, to done one of the experiment, just remove the C-terminal half And in that way, only N-terminal remain the ribonuclease catalytic domain. You can see here the filamentous associated with the anaerobic condition is disappear. So this is just to tell you, yes, the degradosome complexes is really involved in these filamentous changes under anaerobic condition. So then we would like to know, is that only one of the components, or it is every component? So as I told you, we had construct one control strand that deleted at 920 amino acids deleted internally. And this is phenotypic similar to wild type. So this is a control is okay. But with this 800, this is an enolase free degradosome strand. You can see the filamentous shape is no longer associated with the anaerobic growth condition. So that's to say, enolase present in the degradosome is required for this response, okay? And so if you take away the enolase, as you know, enolase is essential when you have a glucose as a carbon source. So we can provide like pyruvate as a carbon source. And also observe that enolase is required. So enolase is required to present in the degradosome to have this morphological induction anaerobically. So also another observation we discovered when you use membrane dye to look, and this filamentous cell have very limited septum formation. So we then ask why is that? So as you know, this is really famous, know that 3 essential cell division proteins, FTSG, FTSA, ZIP-A, they are required for septum formation. Now, with Western blotting shown here, you see among those 3 enzyme division protein, only one, that is the FTSG protein, decreases in response to low oxygen. Now, the mRNA of the FTSG are same. So this is to suggest that this decreases of protein is under post-transcriptional regulation. So let me briefly introduce to you FTFG. Their translational regulation is through a small RNA called DIG-F, and it's in a DIG-F HAQ regulation manner. So you can see when this ribosome binding to the messenger RNA to initiate translation actually is inhibited by the DIG-F HAQ complexes at the entry site. Therefore, by binding to it, they inhibit the translation of the FTSG. So it's easy or even straightforward for us to ask, is this filamentous association under anaerobic is dependent on DIG-F, dependent on HAQ? If that is, then we can link further to the next stage. And that is, yes, we found those are dependent on those 2 proteins, as you can see here. This is another strand. When we deleted DIG-F, the FTSG should decreases, and the anaerobic is no longer decreased. So in this condition, as you can think of, the cell morphology is become rod-shaped, right? If you compensate with DIG-F overexpression, and again, that decreases the FTSG. Similarly, for the HAQ and DIG-F all require. So if you remove the HAQ, DIG-F is degraded because it's protecting it from degradation, and the FTG can synthesize, and you got high level again under low oxygen. So those 2 are required for the reduce of the FTG, and thus required for the filamentous shift. As you can see here, that is no DIG-F, the FTG would be up, and the E. coli would become rod-shaped. Then you increase the DIG-F, and you have a phenotype like this, and similarly to HAQ. Okay, so this is a conclusion. Both siRNA, DIG-F, and HAQ unnecessary to induce cell filamentous under anaerobic growth condition. And since the level of the FTZ decreases under this oxygen limiting condition, and therefore one could assume that the level of this small RNA, it should be up, it should be more. So that is the next, since it's relevant to ribonucleus E as a functional role. So then we look at the northern blotting to detect this small RNA, and this is again use the aerobic, anaerobic, and aerobic alternating conditions. As you can see here, at the low oxygen, when the FTG is low, and you anticipate the mRNA should be up, and that the small RNA should be up, and that is the small, about 53. Big RNA is accumulated, is more in here, right? So also, in order to form this 53 nucleotide, it requires processing. So we look into the precursor of this 53 nucleotide. We found there are AU rich before the 53 functional domain. So we anticipated that is the site required for RNAseE cleavage in order to produce a functional 53 siRNA. So we engineering a mutant replaced with a GGG instead of AU rich. And as you can see here, when you mutate the RNAseE cleavage site, this anaerobic under condition, you can see because you will no longer function, and this E. coli is become rod-shaped. It's like it's no longer, this has no function, and this has function. This under anaerobic condition accumulated when elongated, but this is not. Okay, so they're all okay. So what stage it is a cut? So it's affect the processing, affect the stability, both of them. This I will show you. So this is the stability of the rifampicin. You can see here, when enolase is free, this is no enolase in the degradosome, those 53 DF turnover is much faster compared with the wild type. And because turnover is very faster under anaerobic condition, and that allow the protein to be able to translate it and to be able to do cell division and to be able to maintain at a low condition. If you had the mutant and the mutant become small rod shape instead of filamentous. But in the wild type, that low stabilized RNA, they inhibit the FTZ protein synthesis and become elongated. So that is good. Everything we figured out, and we want to know whether this stabilization of DF under anaerobic condition is specific or is to all the small RNA. And it turns out it's selective, it's very specific for DF. As you can see here, if we use different RNA like SPO 242, this RNA stability in all three strands is very similar, unlike the mutant strand selectively degraded RNA. So this result indicates that the machinery is very selective, very specific, and we don't know the mechanism. And that is really still another problem that needs to be worked out. So as I told you at the very beginning, we would like to know what is the membrane association has to do with this E. coli growth under anaerobically. So this is to show you we want to do imaging. And I remember last time I gave a talk, at that time we just developed the imaging system under anaerobic because the normal fluorescence probe, it won't give fluorescence in the presence of oxygen because they require ATP for protein properly. And this is the protein, it's called EvoGlow reporter that was able to fuse to any protein and they was able to detect at the low oxygen as well. So just focus on this panel. You see the chromosome link ribonucleus E with the BS fusion protein. There are ribonucleus E at the aerobic condition in the presence of oxygen is all in the membrane, just like is a membrane association to the membrane. But if you induce or grow on the anaerobically, you see the distribution is now changed in comparison with this aerobic growth. It's become diffuse form, right? Some are in the membrane, but majority are diffuse to it. So the condition is really changed under anaerobic condition, all machinery. You also can see the enolase, they are similar, has similar, and this control strand also like wild type. However, the mutant strand, which is RNase-free strand, they are still remain in the cytoplasmic membrane. So enolase present in the degradosome determine ribonucleus complexes localization in response to low oxygen. That is quite remarkable. It's almost like oxygen, the enolase may serve as a sensor like oxygen level. And how it happen is oxygen, is enolase iron protein that can sense oxygen or something else. So that is still open question, we don't know yet. But with this kind of association, then you want to know whether the filamentous bacteria that resulted in the change of the protein distribution, right? So we use different chemical that can induce E. coli into filamentous shape under aerobic condition. And with that, you can see all the component RNAseE and enolase, they are similar. There has no distribution change by the by this kind of filamentous shape of the E. coli. So this is to say the diffuse localization pattern of RNAseE and enolase or complex with helicases is specific to anaerobically induced filamentous shapes. So I come quickly to this part for conclusion that E. coli is very smart at this to use the enolastic-degradosome to respond to oxygen and alter cell shape. Now, we of course only answer one of the at least one function of the why the degradosome of E. coli contains the glycolytic enzyme. But our data actually is provide a mechanism that change shapes between rod and filamentous structure under micro anaerobic or anaerobic condition. And as you know, this kind of change is really quite important for UPEC E. coli that cause many women diseases in the urinary tract. These bacteria, when during their pathogenesis, first step they have to get elongated. And since this bacteria contain RNAseE, it's the same family and all the interacting protein, it could be that is provide a mechanism of how that during the pathogenesis of UPEC, they got elongated, and maybe the enolase has a role for that, okay? So, as I mentioned, this is filamentous has that's linked with that. But if you look at the pathogen bacteria genome through the biopsy, and those are the pathogenic strands, very, very bad strand, all E. coli, all in the intestine. And this is the UPEG. And UPEG has 2 copies of the DGF. Then the bacterial strain I have is 1 copy. So those filamentous structure could be used the same mechanism. And furthermore, as I mentioned, those interacting proteins are generally found. And so the discovery we have has provided explanation for that. I want to come to an ongoing project, a new unpublished result. This is just the basis that we publish it. I want to mention another property of this mutant. This mutant strand is not only the morphology on the anaerobic condition is remain rod shape. The doubling time is very fast. If you compare, this circle is the mutant strand. This is the wild type strand. So you, under anaerobic condition, the growth rate, this is much faster than this strand. So it's, in terms of cell division growth rate, so the mutant of this strand, enolase free strand, is fitness. Is really, turns out if you consider the bacteria in the intestine, the fitness would be good for niche colonization and survival, right? So therefore we start to look for human strand, intestine strand, to see whether the morphological and the anaerobic condition, what it happened to it. And since with this background I already mentioned, and so we got one of the strand, as I mentioned to you. This just to briefly tell you that how many E. coli friends we have in our guts. And they are our good friends. If they are good, you don't get sick. But anyway, we would like to know what mutant has to do to this gene relevant to the probiotic strand. So this is strand. 100 years discovery, the NISO has been well established. So when we obtain this strand, and we have this strand, this is very strong, because at that World War I, the German soldier, only one, he is still strong. And this is the E. coli from his face to isolate it, and by the name, this doctor. And so since that, now it's well established as a probiotic treatment for the intestine diseases. So we obtain this strand, and we look, and we grow them under anaerobic condition. As you can see here, this strand under low oxygen is remain as rod shape. It's no filamentous formation. And the division time is very fast. So that makes us to think about whether the gene, the mutant gene, has expressed gene that is required or similar to the probiotic properties like fitness. So we perform RNAseE deep sequence analysis. And this is just to show you, because for this RNAseE, I know it's very sensitive. So sample preparation is very critical. So we had used wild type, mutant, and many biological repeat, 4 to 5, just to set up the established wild type, and have a quality control, primary to look at the 23 sRNA and 10 siRNA that maintain a good 2-to-1 ratio, and then do deep sequences. And then each strand, biological repeat, that we compare among them to show their correlation is higher than 0.1. And so we know those data set are pretty good and reproducible in terms of biological repeat. Then those data, you can do clustering very reliable. And you can see, you may not be able to see clearly, but however, the genes that got upregulated in this enolase-free E. coli strain upregulate the gene cluster in iron acquisition, in pH response, pH acidic, because that's all pretty much in our gut environments. They are upregulated. And also the P1 pili, the FIM-A P1 pili family, and that is important for UPEC, particularly the UPEC E. coli, for initiation of host adherent interaction to establish their initiation for survival, and then later on for pathogenesis. Okay, so that is to say, this enolase-free, the enolase-free strain, turn on many of the genes to do with the cell fitness, and that's to say that it's a strong bacteria, just like the NISO strain. In addition to that, there are many small RNA. We have looked through all the small RNA they are published, and also some new sRNA. And they are, if you compare with the mutant, this is just continuous. This shows you 50. but two of them are obviously decreases in this mutant because this is a lower, this is higher. And those two are responsible actually for the two small RNA affect with whole bunch of the membrane. And they are lower in here, meaning the two component system are less, and meaning this upstream transcription activation mechanism is affected by the enolase. They are not in the RNAseE. And we still don't know the mechanism. And those have been confirmed. You see the two small RNA, they are really decreases in the mutant strain. And they affect the protein. So those proteins, when this is downregulated, they can, because they inhibit the translation, then when they are not there, the protein are more. So they are all confirmed by the mechanism. So we really open up more questions. So all the yellow to show microarray and Western, they are being confirmed. And then the red color is only have the northern and microarray RNA-seq data. Those are called the coli, the type 1 fibrium, the coli, and then the membrane and the biofilm formation. It looks like a strand has a lot of properties that are ready for pathogenesis, or they are used that they are stronger if they are grow under the human intestinal system. So I actually has opened up more questions. This is the degradosome near the inner membrane when RNAseE, enolase is dynamic, right? Sometimes in this form, sometimes in this form. It depends on the extrinsic signals or even the internal, and we don't know how this has been regulated. And if you read my paper, the enolase itself is in the membrane, because this is very interesting how enolase can in the cytoplasmic membrane. It go along with the RNAseE to the cytosol diffuse form, but once the binding site in RNAseE is deleted, enolase in both conditions is in the cytoplasmic membrane. So there are enolase partner that help enolase to be able to locate it in the ribonucleic, in the cytoplasm. It's not just ribonucleic acid. And also, I show you those complexes more active. So the small RNA get degraded and get processed faster. But how about to mRNA, to ribosomal RNA, and to the strand that nasal strand, whether those strand has some similarity in certain probiotic properties. So before I came, I actually had submitted a grant to simultaneously to study nasal along with this mutant. So I come to the end to thank the lab member. So right now, with my PhD student, she's continuing to work on this two-component system, how enolase absence in the degradosome has function on that. And Oleg, who is working on the paper I reported, and he is now like to move to the nasal and to see what's going on. And then Ashan, he would like to work on the female to do with the mammalian cell to establish the adherent interaction, how this strand can provide fitness during this interaction with mammalian cells. And I think that's good for him to be able to have cross-disciplinary function, the research program. Okay, so I cover you a little bit introduction and enolase present in the RNA degradosome and its function. And so I show you some anaerobic phenotypes of this mutant like that of NISO 1917 used for the probiotic treatment. And then the ongoing research direction have shown some preliminary results. And there will be function of E. coli enolastic-degradosome in cell fitness and host adherence under micro anaerobic condition, and function of enolastic-degradosome in probiotic properties of ECN. So there are some two sentences that I would like to share with our young students. By giving this talk, right, of course I have to thank my former mentor, Hans Bremer and Sten Cohen, and all those great scientists that they published good papers. I was so interested in reading it, and then I ended up in my career based on their discoveries. And so I really thank them. But those are the two I want to say. For me, my personal experiences, scientific experiments often answer one question and open a new question, even a new field. And the process is often complicated and painstaking, right? As we all know. But it is worth of it is done, because you know that what you did, and you know that it make you as your career, and with that you can train more young people, and with that you interaction with people, and with that you connect to the people globally. They don't need to know you, but they know your work. I don't know Tomizawa, I don't know David Aperian, but I know them through reading their work. So I really think, but not worth doing scientific experiment already know the answer. Because I sometimes students came to my lab and tell me that that's what I do. And I said, why? He said, because this is safe, and I know they will get results. And I said, that's not worth your time. And that is my advice I really want to give the student in this audience. And thank you for be patient. And thank you so much. Namaste. I should say, let me say, Tanyawa.
Thank you very much for the complete story that you have narrated about the enolase. Maybe it is close to lunchtime. We can have some quick questions. Yes, Sarmishta.
Thank you so much for the talk. E. coli still continues to surprise us. So about this enolase, the one which is in bound state with this degradosome, was it catalytically active, or do you think there will be two different population, one which is catalytically active and participating in the glycolytic cycle, and this is the one which is?
Very good. We purify enolase with degradosome with enolase. We did some of the in vitro enzymatic catalytic, and the enzyme activity is there. Okay, of course I don't know. In E. coli, the enolase has bound form and free form, and I don't know which form actually participate more in glycolysis. But the enolase free strand tell me when you have more free enolase, the carbon source, this metabolism, this central metabolism pathway get up. Okay. I did not say the data because I do cluster RNAseE. So somehow, I think when it is in the degradosome, they probably function more as a regulatory to the ribonucleus E. Maybe also have some contribution to the glycolysis. But when it's free from it, it seems really can do more on this glycolysis and leads to TCA cycle.
The residues which are residues of the enolase which binds to this
don't
are
know.
different. Oh, you do not.
Don't know.
Okay.
Yes,
Okay.
don't know.
Thank
Because
you so much.
it's hard to do
It's hard to
mutagenesis on the enolase part. I just don't know. I wish I can have.
But it
such
can be modeled or something, because then you will know whether it's falling into catalytic space or
I
not.
had asked actually some structural biology try to figure it out, and so far they didn't tell me. They told me can be, but they didn't tell me where. But maybe someone that if you're really interested in this, provide me some hint, I would be happy to know and do some mutagenesis.
Wonderful. Thank
There
you
has
so
to
much.
be glycolysis still
Naresh.
functional. Yeah, that is really good. That is the enolase side. I really want to do it.
See, enolase has been shown, and at least in yeast, is involved in the RNA transport, tRNA transport into mitochondria. One of the enolase is the component
know.
of that.
Yes.
And other thing is, do you see any structural alteration in enolase in the presence of oxygen and anaerobic condition? Because we have shown some proteins get dimerized in the normal condition, oxygen condition, they monomerize. Is there any, did
Yes.
you look at the structural aspects of this?
Okay, you're talking about FNR. Yeah, FNR, there is oxygen present or not bind, and in response to the protein dimerization activity. So we have established a system because you want to do structural work to be able to snap, freeze, enolase, isolated, remain anaerobically. So all the purification protein system has to be different from regularly. So that is the OLEG that he is doing it. And then after, because the structure has been solved for other, like you said, Easter, and those are the protein regularly isolated in the presence of oxygen, and that's very quickly. So we try to figure it out. We actually already use this chamber we established, have the protein purified from there, and we try to whether we can maintain something and then quickly to do structure. And hopefully we can find maybe iron, something binding site in there. There actually has one bacterial strain from anaerobic bacteria published by, not published, it's only abstract by Japanese. And it says the enolase bind to iron. But that is only maybe 300 words abstract. They didn't publish, and I cannot follow up with it, where is the safe. But maybe by prediction also be able to know where maybe the iron binding site. So that is very important. I think enolase is getting very important, particularly the cancer. Those people in hypoxia, they actually switch the metabolic mode to use enolase as a function. And that is low oxygen. So I'm very interested in that.
It's a very good talk. Here, I am here.
Thank you.
So I was wondering, actually, the extension of those two questions, adding another dimension of membrane biology, anaerobics versus aerobic. Instead of eno-free, have you tried enolase inactive degradosome?
Would you repeat your question again?
You have tried eno-free degradosome.
Okay.
So if you have inactive, then will it be associated with the membrane and
Enolase inactive
degradosome?
But
So instead of leaving the enolase,
okay, I got you. You're saying that do I have inactive enolase bound to the RNAseE as a degradosome and ask where is the complex distributed? Yeah, the enolase is essential. So far, I think the deletion need to provide with carbon source. I am not aware whether there is a catalytic mutated enolase enzyme. I should look. It may not exist, but I should look. But I probably can do catalytic domain and mutated. So you are talking about whether catalytic activity of enolase affect the subcellular distribution under anaerobic condition, right? Because
no, I am not asking that. I was wondering, because of enolase is a part of the degradosome,
right,
it stabilizes the degradosome
right,
as a component. May not be activity. Then the change in the membrane biology during anaerobiosis, maybe the association is better when you have an enolase, whether it is active or inactive.
Yes.
So the membrane biology, membrane physics, membrane composition would change anaerobiosis and aerobiotics.
Right.
So maybe in anaerobiosis you need a component of enolase where the conformation of the degradosome is well suited to the membrane, and it keeps it.
Possible. Yeah, that is good. Hypothesis. Another thought on that. Change the conformation that fit into with the membrane interaction. Sure. Yeah. Conformation per se. Yeah.
Thank you.
Thank you. Thank you.
Okay, maybe you said this and I missed it, but does it act as a sensor or something on the plasma membrane? Why is it association with the plasma membrane so important?
Well, membrane is everything linked to the life of the cell, because it really is a good signal to compartment. Otherwise, I think if you have ribonuclease everywhere, I think the RNA degradation probably got crazy, and the regulation will be more difficult. But in that way, at least one more easily is to regulate the conformation, because the membrane has a conformation effect on the protein activity, right? And the whole thing is all in one spot. And all you can do, they all connect to each other, and then they're working quickly. And it turns out it's very efficient. And I think for bacteria, the genome is small, and the protein are so much and so many messenger RNA they have to work on, then it's only ribonuclease. I think it's better to do it. So far, because we have found the ribonucleic integration site is through charge and also has a small peptide they tether to it. So it's not possible to make the really good protein that not bind to the membrane. And so your answer is difficult, but good reason that you can think of, but really to prove it is hard. But the dynamic, the ribonucleic acid on and up, that is in real life. And that has to do with the conformation. I like that model because when you conformation and change it, the activity also regulated. And that is a good way to think about it. And I like that, your idea.
Professor Danan.
DIC
Is phage encoded RNA, is it correct? Prophage encoded RNA?
Yeah, it's a phage, it's a prophage encoded, but all the strains have many prophages.
Yeah,
I
but
don't know.
how this link of expression of DIC gene and your hypoxia condition, anaerobic condition?
What is the link between?
Expression of DIC and anaerobic condition.
Okay, in the wild type, E.
Yeah,
coli, right?
yeah.
On the anaerobic, the DIC got stabilized. So you have more DIC,
Okay.
DF, the small RNA, you have more. The prophage encoded transcript, they got processed, but the small RNA is accumulated. And this is inhibit FTFG, and that's why the E. coli they don't get. Yeah, so it's stabilized. Now, bacteria, the pathogenic bacteria have many prophages. And started earlier time, they come as a stable genetic elements. So that, I think they are talking to each other. So enolase is really a surveillance, like, to sensing. Or for E. coli site, they want to escape from the dangers and they just turn on everything for survival.
Hexokinase is also in two forms, cytosolic and membrane. Hexokinase,
Yes.
another glycolytic enzyme, cytosolic and membrane bound enzymes, specifically in cancer cells where the glycolytic rate is much higher. So here also enolase in the two forms is regulating glycolytic pathway more.
You get into the question that many people are asking me. In mitochondria, right, in those in cancer, they really cytosolic glycolysis become predominant, and this enolase actually is encoded by the mammalian host. It's not mitochondrial protein, right? So also kind of two form, and also they have RNA binding activity in mammalian cells. So bacteria, in my studies, I cannot differentiate bound form, two form. Do they have any role played differently? But if I have a mutant that can only establish as because I believe free form has glycolysis ongoing, but that's something for the future.
Yeah, that would be maybe last question.
Nice. I have two questions.
Hi.
One is a bit general one because this is an extension to Sharda ma'am's this thing, but where the enolase has been attached to the membrane. So have you seen any changes in the phospholipid the interaction? Because in the membrane, when there is an aerobic, when the aerobic conditions change to anaerobic, or you use the potassium chromate, there may be some changes in the phospholipids because of which the enolase is getting specifically bound to the phospholipids, which has been removed in case of anaerobic.
You give me another direction.
Another perspective on this thing. So
Yeah,
whether
the
there is
lipid
a change
is
in
very-
phospholipids causing
Yes.
this specific change rather than
Yeah, you give me very good direction. This is something I have to think of, the lipid modification, particularly on the different environment. That lipid also have different modification, and they could well be one of the reasons play role with enolase, and then indirectly to RNAseE, to change RNAseE conformation. And that is actually the hypothesis that I didn't really clearly But I think your question now make me think even more clearly about that maybe is the case.
And second thing, ma'am, the link between this change in shape, how is exactly it's linked with the RNAseE? Because you have proved in steps, but is the correct mechanism still you are still working on it, or it's there?
Okay. That only when I work on ECN coli, then I will know, because ECN on the anaerobic is very short, rough shell, right? So if I think that ribonucleus E and the thick F is the sole reason, I should be able to engineer the strand to see they get elongated. So I have no answer right now. But for our work, the establishment was the ribonucleus E get decreases, and so the thick F got stabilized, and they inhibit the FTZ, and that the septum formation has become very rare event, so they got elongated, right? But I need to use ECN different strand to prove whether this is the only reason. It could be other genes that can play a role, but I have to discover. But use ECN, I should be able to find it out.
Thank you.
Thank you. Good question. Very good.
Okay.
Okay.
Thank you very much, Sue Lin-Chao.
I'm very happy.
Yes. And on behalf of the university, let me have the pleasure of honoring you with a small memento from university.
Thank you. Like this?
Yes.
Thank you.
Yes. This is the memento from the university.
Oh, thank you.
Yes.
Thank you. Thank you.
Yeah, please sit. Prakash?
I said again.
Professor Prakash will present a vote of thanks.
Respected Vice Chancellor, Professor Appara Purley, distinguished speaker, Professor Sue Lin-Chao, Dean, School of Life Sciences, Professor M.N.V. Prasad, and faculty colleagues, students. It's my privilege to propose a vote of thanks in this occasion. I sincerely thank our Honorable Vice Chancellor, Professor Appara Purley, for his constant support and readily accepting to host this distinguished lecture by Professor Sue Lin-Chao when I proposed it. And on behalf of University of Hyderabad and on my personal behalf, I sincerely thank you for your excellent talk on enolase and its association with RNA-degradosome complex. Professor Sue Lin-Chao for delivering such a wonderful talk today. And I thank Dean School of Life Sciences, Professor M.N.V. Prasad, and all heads, 4 department heads, heads for their support and encouraging students to actively participate in such a lecture series and also this current lecture. I thank PRO office for making all the necessary arrangements and especially responding timely and quickly for organizing this event. And I thank all the faculty, students for attending to this lecture and thank you one and all. Thank you, Sue.